It's a great pleasure to be here and to talk to you, Dr. Sokoloff, about your life and your work in the field of neuroscience, particularly with regard to metabolism. I was looking at your bibliography and noticed that your life has basically been in three areas. First of all, you started with the Army, then you moved to the University of Pennsylvania, and then at NIH ever since then. You graduated from the University of Pennsylvania and you joined NIH in 1953, but although for a while you overlapped between Pennsylvania and NIH, you've been editor on many boards of journals, particularly the Journal of Neurochemistry, the Cerebral Blood Flow and Metabolism Journal. You've won many awards, including the Lasker Clinical Medical Medal for the Research Award, 1981, the Carl Lashley Award of the American Philosophical Society, and most recently the National Academy Sciences Award in Neuroscience and the De Hevesy Nuclear Medicine Pioneer Award in '88. You have over 320 publications in this long and productive career of yours. And my first question is, how did you get started in this particular field? What were the early thoughts that got you started in this area?
I graduated from medical school at the University of Pennsylvania when the war had just ended, this World War II. And at that time, internships were rotating internships. And we were on an accelerated program where all the requirements had to be completed within 9 months. In this rotating internship, I started off with psychiatry, and by the end of 9 months, I completed my requirements for the rotations. And then they decided to go back to the regular one-year program, which meant there would have to be an overlap of internship classes. And our group, our class, was given another 6 months, so we had a 15-month internship. The requirement that they made was that the final 6 months, the extra 6 months, would not be rotating, and you would be serve on only one service. And I was given psychiatry to serve on. So I had 6 months of psychiatry in addition to my previous 6 weeks that I'd had earlier. And this was at a time when physicians were coming back out of the Army and out of the military forces and taking postgraduate training. And so the Philadelphia General Hospital, where this occurred, became sort of a training center in postgraduate psychiatry, among other things. And so I got quite a bit of intensive training and education in psychiatry. And because of that, when I went into the Army as a medical officer after my internship, the Surgeon General of the Army of the United States ordained me a neuropsychiatrist. And for the next 2 years, I did psychiatry. It would not have been my choice. I was, even then, I was interested in basic science. I was interested in physiology. But that's what the Surgeon General ordered, so I did it. And I practiced psychiatry as I had learned, although I had a lot of reservations about many of the ideas. Freudian psychiatry was then predominant. Well, in the course of the 2 years that I was there practicing the psychiatry as I was taught, I had a patient that got well. And I was so shaken. I didn't understand how her talking and my listening would make her better. But I became interested in how the brain works. So when after 2 years, I left the Army, and I went back to the University of Pennsylvania, where I knew that Seymour Ketty had been working with his newly developed nitrous oxide method for measuring cerebral blood flow and metabolism in conscious men. And I thought that technique would be very useful for studying how the human brain normally functions and what goes wrong with the brain and various neuropsychiatric disorders. So that was why I came back to the University of Pennsylvania, and I became interested in the blood flow and energy metabolism of the nervous system, and been staying in that in one aspect or another ever since.
Yeah, ever since that time.
Right.
I noticed that really right from the very start, and this was, I think, the early 50s when you began, basically, that what you were getting into was the term that subsequently I think you developed first, which was People were calling it quantitative autoradiography. You called it metabolic encephalography. And I think it's a good term because it describes the fact that we're looking at a function. It's often misthought is that this is looking at a structural change. It's really looking at the function like an infrared photograph. And yet it's interesting that it seems to coincide with the structure, with the anatomy. Although it's not precisely so. You might want to explicate that a little bit for those who are not familiar with this technique.
There were some neurophysiologists or neuroanatomists who I think were misled by seeing illustrations of sections of brain that looked to them almost like stained sections. And you could see changes in certain areas in different functional states in these images. So they called it a neuroanatomical technique. Well, as a physiological chemist, I didn't like that.
Exactly.
It was not an anatomical technique. It was a dynamic process that was being looked at. In fact, it was rather interesting that in one paper, one of our leading neurophysiologists had mentioned that it was rare that a neurophysiological technique, and he meant electrophysiological recording, would precede a neuroanatomical technique in uncovering something about the brain. And by the neuroanatomical technique, he meant the deoxyglucose method. Well, in a subsequent paper, I wrote that metabolism is just as much a physiological process as electrical activity, and that in fact the deoxyglucose method or the local blood flow autoradiographic methods were looking at the dynamic process, and it was just fortuitous that the images were so similar to the anatomically stained sections. All that showed was that the neuroanatomists all these years hadn't been doing something irrelevant, that there was a concentration of function in specific structures, that the anatomy was the base on which the functions were distributed. And that was fortuitous. It didn't have to be that way.
It was a happy development because it tied together the anatomy and physiology in a way that electrophysiology alone was not doing with the neuroanatomy.
It meant that the functions are really segregated into specific anatomical structures, and it isn't just a random network of things happening.
I think that was a very important step. I think the initial work you did was on the whole brain metabolism. You were completing a number of studies on that, but then very quickly you developed the regional technique.
Well, what happened was that when I came to work with Keddy, there had already been a number of studies with the nitrous oxide method, which measured blood flow and metabolism of the brain as a whole, not locally. There have been a number of studies with that method in which oxygen consumption was reduced, but there had been none in which oxygen consumption had been increased. In anesthesia and in various kinds of coma, metabolic activity was depressed. Now, the first project actually that I got associated with when I came to work with Keddy was to look for some condition in which the oxygen consumption of the brain might be increased. And we chose hyperthyroidism, thinking that in hyperthyroidism, all the tissues have an accelerated energy metabolism, and the brain would be one. Well, it turned out that even in hyperthyroidism, there was no increase. And hypothyroidism and myxedema, there seemed to be a decrease. Then we studied slow-wave sleep, and we found no change in oxygen consumption. We studied the performance of mental arithmetic, and we found no change in oxygen consumption, and in fact, of glucose utilization either. And so that was a dilemma. In all structures in which studies have been made of this type, there's always been a good correlation between the amount of work that the tissue does and the amount of energy metabolism. Why would the brain be different? When the brain does more work, you'd expect more energy metabolism, and yet we weren't finding it. Now, one possibility that had been suggested by a number of people was that the reason you couldn't increase oxygen consumption in the brain is that the brain's energy metabolism is already at a maximal rate and can't be increased. That's why you can only find decreases and never increases. Well, that didn't sound sensible to us. That would be a foolish thing for nature to do. That would be like somebody designing an automobile where the motor always goes at maximum speed, and the way you change the speed of the car is to just gear down. That's not a very sensible way to design a system. And nature's had 4 billion years of experience to not make mistakes, to correct mistakes like that if it had made one. So we had to look for an alternative possibility. And the alternative possibility that we considered was that the measurement of the average of the whole brain was the wrong thing to study when you're trying to relate it to functional activities which are confined to specific regions of the brain. In fact, they may be confined to regions so small that any change in that local region would be diluted out by lack of change in other parts of the brain, or even reciprocal changes. And so we thought the way to attack this was to get methods that measured local energy metabolism. Well, it seemed like a big order. But Seymour Ketty and Bill Landau, Walter Freigang, and Bud Rowland and I finally worked out a way for at least measuring local blood flow that was based on the principles of inert gas exchange that Ketty had developed earlier. But we needed localization. And the method was based on the assumption that we were dealing with a homogeneous tissue. Dissecting out the brain portions never led to homogeneity. We always got mixed tissues. And so the idea of using a quantitative autoradiographic technique, which would get us a little closer to homogeneous tissues, occurred. And out of that came the quantitative autoradiography. And the local blood flow method was developed by this same group of people in the 50s, middle 50s, about 55, 56. And then after that came the idea of adapting that autoradiographic method for measuring local energy metabolism. Because the radioactive isotopes of oxygen are so short-lived, it wasn't possible to do an autoradiographic method with oxygen. But fortunately, in nervous tissue, except in ketotic states, glucose is almost the sole source of the oxidizable substrate, the sole substrate for energy metabolism, for oxygen consumption. So that if you measure glucose utilization, it's just as good as oxygen consumption. And so we first tried to do it with radioactive glucose, using radioactive glucose as a substrate and trying to measure the accumulation of radioactive products in the tissue, which we can measure autoradiographically. The problem with that is that with glucose, the carbon is converted too rapidly to carbon dioxide, which leaves the brain quickly. Or if you use glucose labeled with tritium, the tritium would be converted too rapidly to tritiated water and come out too fast. That meant that you would have to work very, very short times before you lose any product to be able to get a quantitative method. Well, if you work too short a time, then you don't know the specific activity of the precursor molecule in the tissue from measurements in the plasma. And so it was a dilemma. If you go too short, you don't know the precursor specific activity. If you go too long, you've lost product. And so I set the project aside until sometime later I became aware of this interesting compound, 2-deoxyglucose, which gets metabolized just like glucose up to a certain point, and then the product gets trapped for a reasonable amount of time. so we could go long enough time to be able to get a reasonable estimate of the precursor-specific activity without losing any significant amount of product. And out of that came the local glucose utilization method. And that showed, in fact, that there was a close association between energy metabolism of the brain and of brain tissue and the functional activity within those portions of the brain.
I think it's true to say that until you developed the deoxyglucose method, although there had been good support with the nitrous oxide method about the correlation between cerebral blood flow and metabolism of the brain, that this really tied it down in terms of the regional distribution problem, that there was a adjustment of the brain function in terms of regional metabolism changes that would correlate with the blood flow changes.
Right, yes.
Very precisely.
Yes. There had been some regional cortical blood flow measurements done by Lassen and Ingvar using an inert gas clearance method, but that was only from the cortex, not from the innermost parts of the brain, where with our autoradiographic method we're finding some very interesting effects. Right. There's more going on in the brain than just what goes on in the cortex.
That's very true. Yes, people forget the earlier parts of the brain. Yes. Now, quite very early on, you were concerned about the resolution of this method. And I think you state in one of your papers that the spatial resolution is about 100 micron now.
Right.
200 micron, quantitatively.
Qualitatively, you can see down to 25 microns.
25 microns?
Yeah, but when you measure the concentration locally, you're getting a smearing effect because of the resolution isn't that good. I guess you'd call it partial voluming effects that you see with PET scanning.
Right. The limitation in terms of temporal resolution it can be varied according to the method. What are the parameters for temporal resolution?
Well, unfortunately, the temporal resolution is not very good. The reason we use deoxyglucose is because it allows us to go a long enough time so we get a reasonably accurate estimate of the precursor pool, integrated precursor pool specific activity at the enzyme sites. Exactly. Which means you have to go a long enough time. Yeah. You could go just as short a time with deoxyglucose as you go with glucose, but then your results will be just as inaccurate. Right. So to get accuracy, we have to go a longer time, 30 to 45 minutes. Right. And I don't see any clear way of getting a better temporal resolution without giving up accuracy.
Even with oxygen, labeled oxygen itself?
Well, I'm talking about autoradiographic method.
No, with PET scanning.
With PET scanning, yes, you can go very short times. Mm-hmm. You can measure blood flow within a half a minute with using O15 labeled water. Yes. Mm-hmm. To measure oxygen consumption, it takes a little longer because to measure oxygen consumption, you must have at least 3 scans, one after another. You must have a scan for blood flow, so you have to do a water scan. You have to do a scan for the amount of oxygen that would be still bound in the hemoglobin in the blood in the tissue, so you do a carbon monoxide scan, and then you do an molecular oxygen O15 scan to measure the oxygen extraction fraction. And then out of those 3 scans, each with its own source of errors, you accumulate a lot of data from which you magically compute the so-called oxygen consumption. Yeah. So that takes at least 6 minutes.
The temporal resolution of the technique of quantitative autoradiography, and especially for clinical work, it appears to be difficult to decrease it below 5 minutes. Is that correct?
With the autoradiography and with the particular methods we're talking about, it's very difficult to go below 5 minutes. With other methods, you can go shorter times. In our laboratory, where we use mainly the quantitative autoradiography, not positron emission tomography, where you get only a single time point. Right. When we have to study a condition which would not be constant, we need something that's very transient, very short. we'll switch to the local blood flow method with iodo-N-pyrene, which only takes a minute. And then we do the autoradiography after 1 minute, at 1 minute after the start of the infusion. But with glucose utilization, you have to go longer. With positron emission tomography, again, if you use fluorine-18 fluorodeoxyglucose or carbon-11 labeled deoxyglucose, you have to go 30 to 45 minutes at least. But with the blood flow, with oxygen-15 water, or with the oxygen consumption method, you can go, well, with the water, you can go less than a minute. For oxygen consumption, you need these 3 scans. And with the 3 scans, it'll take you up to 10 minutes at least. It'd be very hard to go less than that.
Those are the limitations. I tried to get some impression by reviewing your publications of the various aspects that you've contributed to, and it seems to be an enormous list, because what has happened is that with this technique is so fundamental and basic that it has been used in many fields. And apart from the basic questions of mapping the metabolic functions in a quantitative manner, this has contributed to the neurophysiology, neurochemistry of consciousness and behavior, unconsciousness, coma, answers to the energetics of the brain, mapping functional states in the brain, and application to disease states, particularly Parkinsonism. You've studied the MPTP model, which is, we've shown also the effect on the, and then also in development and aging. These are a large list of things that I'd like to pick out 3. One is the brain energetics as one approach to the problem of, on your earlier work, it looked like the brain was a constant consumer of energy, very hard to disturb. It was easy, as you said, to decrease it, but very hard to increase it, except regionally. But even in the regional, if you add it up, it was remaining remarkably constant. Secondly, the application to disease states, particularly the Parkinsonism work and the aging work. If you could briefly touch on those 3 aspects.
Well, the energetics aspect of it has been very interesting to us. I earlier had said that we thought it would be foolish for the brain to do more work and not use more energy to support it, that all other tissues that do work use energy in proportion to the amount of work they do. The question did occur to us, what is the work that the brain does? We know what the heart does. It pumps blood against the pressure head. We know what the skeletal muscles do. They pull up weights against gravitational forces. That's physical work. The kidney does physical chemical work. It absorbs substances against osmotic gradients. That takes energy. Energy has to be conserved, and it's converted from one form to another. But what is the work that the brain does? We weren't sure. Well, and then once we found that, in fact, there is an increased glucose utilization in specific regions of the brain in which the activity has been increased for whatever reason, then we raised the question, why is the glucose utilization increased? What is it, the energy going for? Where is it going? And so we embarked on a series of studies which first showed that the glucose utilization was more or less proportional to the spike frequency in the terminals. Well, that was a first start, that it was related somehow to the spike activity. Well, what's a spike? Well, a spike is the result, Hodgkin-Huxley theory, a spike is the result of sodium entering the cell and potassium coming out. And resulting in increased extracellular potassium and increased intracellular sodium. Well, neurochemists have known for years that when they want to increase oxygen energy metabolism of a brain slice in vitro, you just add more potassium in the extracellular fluid and it stimulates the metabolism. And so we thought maybe that's what's going on. That each spike puts out a little bit of potassium, takes in a little sodium. The extracellular potassium goes up, the intracellular sodium goes up. Now, what does that do? How does that convert to energy metabolism? Well, there is this sodium potassium ATPase enzyme which is involved in the active transport of potassium back into the cell and sodium out of the cell to restore the ionic gradients. So therefore, we thought maybe what's happening is that spike activity causes dislocation of the ionic distributions, which turns on the sodium potassium ATPase, which splits ATP, and biochemists have all the mechanisms. Once you split ATP, energy metabolism will be increased. And so we did experiments there where we found that increases in glucose utilization associated with the stimulation of functional activity could be blocked by specific inhibitors of the sodium potassium ATPase. And that any means of which we could increase the intracellular sodium or extracellular potassium by using compounds like veratrogin, which opens the sodium channels and lets sodium flow in, would stimulate glucose utilization, and you could block it with Wabe, which blocks the sodium potassium ATPase. So we, I think, solved the question: what is the work of the brain? What is the energy being used for? Now, an interesting historical thing comes out of this. What are we looking at when we look at the changes in glucose utilization associated with functional activity? We're not looking at the functional activity itself. We are looking at the consequences of the functional activity, and we're looking at the energy consumed to undo the consequences and bring it back to the normal state, which brings us back to what Gerard and A.V. Hill and others used to call it the heat of recovery. We're looking at the glucose utilization is the heat of, it goes into producing the heat of recovery associated with restoring conditions back to the pre-activity level, which is interesting. So that the energetics are interesting. Now, what were the other?
The application to disease states, such as the MPTP model.
Yes, that was rather interesting. From histological or pathological examinations, it was known that MPTP knocks out the substantia nigra zona compacta, and it produces a clinical picture in the primates that is exactly like human Parkinsonism. So we knew where to look. But one of the advantages of the oxyglucose method is that you don't have to only look at one or two places. You see the whole nervous system all at once. And so we thought maybe we could learn something about some of the abnormalities, clinical abnormalities that you see in Parkinsonism. We might be able to see in these models by looking at the whole nervous system all at once.
Something you can't do in the human.
That's right. And so, yes, when we studied these animals with MPTP-induced Parkinsonism with the DG method, the zona compacta was gone. But we found a lot of other areas that showed altered glucose utilizations that was used by one of our Hughes medical scholars, one of the students that come here, spent a year here, and worked out the pathways involved in the eye signs in Parkinsonism from looking at the changes in the local glucose utilization. We found changes in the globus pallidus, glucose utilization, indicating that pathway was altered. For me, one of the most interesting things was when we looked at the effects of DOPA. In these monkeys, if you give them DOPA therapy, they quickly return to normal. It's spectacular how effective the DOPA is in reversing the signs and symptoms of Parkinsonism. Now, when we gave DOPA to normal animals, it didn't have any effects on local glucose utilization, or no behavioral effects. But when we gave the DOPA to the MPTP-treated animals, where the clinical picture reversed to normal, but there were tremendous increases in glucose utilization in many different areas of the brain, areas that were not even affected in the were not decreased in the MPTP Parkinsonian state, which indicates that dope is doing something different from what we've been thinking. It has some kind of effect in these in the Parkinsonian monkeys that is above and beyond what is needed to restore the systems. What we're thinking is that it's related to supersensitivity that develops in the deafferented pathways.
Related to the substantia nigra.
Yes, right. But that, for example, the visual cortex showed tremendous increases in glucose utilization when we give dopa to the Parkinsonian monkeys, but no effect whatsoever in the normal. So that has not, that story has not been completely solved, but it looks like there's some very interesting questions to be examined there.
It's very important clinically because we know now that L-DOPA is not the answer. Patients do not get all the symptoms resolved, and this may be a very interesting clue to follow up with regard to the pathways. The third question was, part of that question was related to your studies in development from the fetus and also in aging. There were some applications over there in terms of what's happening to the metabolism in development.
Now, we ourselves have not studied the very young animals with the deoxyglucose method. Dr. Kennedy and Dr. Grave and Jane Jaley in our laboratory, back around 1970 or so, studied local blood flow in the beagle puppies as a function of postnatal development. And they had found that blood flow is low everywhere in the brain at birth, and then rises with time and reaches a peak and then comes back down again and levels off at the adult level. And the peak is reached at different times in different structures. And it seemed as though those structures that were most mature at birth, the peak, those lower in the neuroaxis, those structures, the peak was reached at earlier times than in those that developed later. And we all wondered what that meant. Now, more recently, Chugani at UCLA, using the fluorodeoxyglucose method and positron emission tomography, has been studying development in human children. And the time scale is different, but he finds the same phenomenon on glucose utilization. And has been able to offer a very reasonable explanation that the peak is reached and then comes down again as there is retraction and loss of synapses, selective loss of synapses and cells. We now know that in development you have a surplus of synaptic connections and then there's a selection out and a discarding of unused ones. And that his work indicates that that is the reason why it comes back down again and levels off at the adult level. Dr. Nalick, who's at the University of Nancy in France, who spent a year in our laboratory and learned the method, has gone back to France, and she's been applying the deoxyglucose method to rats in the postnatal developmental period. It's a tour de force for the laboratory skills that we ourselves don't have. We can't study those little tiny rat pups.
And
she's getting similar results in the rat that Chugani got in man.
The one particular aspect of this mapping which is extremely fascinating is the paper you published in 1984 in Science, the metabolic mapping during rewarding self-stimulation of the brain in rats with Perino and the other colleagues. Perhaps you'd like to explain that paper a little bit more, because I think that has some very interesting correlations.
Well, that was related to this fascinating phenomenon that I think Olds first described a number of years ago, where animals will push a lever to give themselves electric shocks in some specific regions of the nervous system. And the question was, why do they do this? Dr. Perino, who's a neuropsychologist, believes that this is a model of studying motivational behavior. Yes. And so she did these studies and with a collaboration with Ralph Esposito, who was there at the time in the lab. He's also a neuropsychologist. And what they did is they placed the electrodes on one side in the ventral tegmental area, and they trained the animals to stimulate them, and they would learn to stimulate themselves. And then the question was, which areas of the brain are involved in this so-called motivational behavior? What they found was that, of course, the pathways coming from the site of stimulation where the electrode is placed, the pathways up and down are actively stimulated because they're electrically activated. So that's not very specific or interesting. But then they looked for areas beyond where the direct pathways from the site of stimulation would be. And they also looked at different regions where the electrode was placed. They also looked at the substantia nigra, another region that supports this stimulation. And they also studied the effects of the investigator-administered stimulation. What they did is rather clever. They trained these animals to self-stimulate, and then they determined the frequency at which they would normally stimulate themselves. Then they allowed this behavior to become extinguished by withholding the electrical stimulation. It's pretty rapidly extinguished. And then they would stimulate themselves, the animal, not instead of the animal doing it, they did it at the stimulation frequency and the current that the animal itself would do. Now, when they did that, that became the control for the motivational behavior. And they found differences. And one of the major differences they found was that the nucleus accumbens was activated bilaterally, even though the stimulation was on one side. The nucleus accumbens was stimulated bilaterally, regardless of where the electrode had been placed, whether it was in the ventral tegmental area, the substantia nigra, or other places. So it suggested that somehow or other, the nucleus accumbens is involved in this motivational behavior, which is interesting because the nucleus accumbens has been suspected of being involved in other conditions. In fact, there's been some theory of schizophrenia being related to nucleus accumbens.
And this is the nucleus that did not show itself only on the experimental stimulation, but it appeared, yes, only with the self-stimulation.
Self-stimulation, right. Bilaterally.
Yeah.
Right.
We were talking about the metabolic mapping in self-stimulation of the brain, and it's a very good example of the correlation between the anatomy and the physiology. And another beautiful example of that is in your work on the metabolic mapping of the visual system, because Mort Mishkin, whose lab I had first worked in when I came here on memory, has been working for years, over almost 3 decades now, on the anatomy of memory. And you did the work on the metabolic mapping, which gave us some very interesting data. Perhaps you can briefly...
Yeah, that's a fascinating story. After we had gone through the theoretical developments and the experimental measurements that we needed to establish the deoxyglucose method, we raised the question of what happens to glucose utilization in with changes in functional activity at the local level. Does it occur? We weren't even sure that glucose utilization would be changed when you change local functional activity. And if it did occur, was the method good enough or sensitive enough to pick it up? So we embarked on a series of essentially what a biochemist would call a recovery experiment, where you do something where you know what the result should be and test your method to see if it picks it up. So we altered functional activity by well-known methods in local regions of the brain and then used the deoxyglucose method. And one of the things that we did was to study in the albino rat the first visual stimulation. So we knew that in the rat the pathways are crossed about 85%, so one should get a lateralization. So we patched one eye and stimulated the other eye with a light, and we didn't hardly see anything. And then it occurred to us, well, these are albino rats. And we later learned that with the intensity of the stimulation we were using, we were blinding the animals. So then we thought, well, if we can't stimulate, maybe we could see a difference if we just patch one eye or enucleate one eye. And so we did that, and it worked beautifully. Contralateral to the enucleated eye, the superficial layer superior colliculus was depressed, the lateral geniculate was depressed, the visual cortex was depressed as far as glucose utilization was concerned, and that was fine. Then later, when we not much later, we decided, gee, it would be interesting to do that in a monkey, see what happens in a monkey. We knew that the pathways there are crossed about 50%. They have a binocular visual system like man. But what would happen if you patched one eye? So we patched one eye and we studied it. We got the autoradiographs. Oh, I have to introduce something else. We wanted to patch one eye and maximally stimulate the other eye. So we asked Bob Wirtz, who's a visual neurophysiologist, what's a good way to stimulate a monkey's eye? And he suggested using a black and white pattern of stripes in all directions to stimulate all the orientation columns or so on. Well, so we had a screen rotating around the monkey's head with this pattern of stripes in all directions while we did the deoxyglucose method for 45 minutes. When we got the autoradiographs out, we looked at the visual cortex, and there were stripes going right through the visual cortex. And we looked at that and said, what is that? Mike DeRosier was at our laboratory at that time, and he came up with the possibility that, suggestion that maybe it's a reflection of the stimulus pattern. And we discussed that possibility until we finally realized that that's something silly, because what would happen if we would wave an American flag in front of the monkey's eye? Would we see the stars and stripes on the autoradiograph? No, that couldn't be it. It had to be something else. And so we asked another neurophysiologist, and we asked an eminent neuroanatomist what they thought these stripes in the visual cortex meant. And they said it's an artifact of autoradiography. So for months later, we repeated the experiment. Both eyes open, no stripes. Both eyes closed, no stripes. One eye closed, stripes. And we did it over and over again, and we also used methyl glucose, which is an analog of glucose like the oxyglucose that is transported across the blood-brain barrier by the same carrier and enters the brain the same way, but it doesn't get metabolized. So it's acts like a tracer for blood flow and transport only and not metabolism. When we use that, no stripes. So it was something related to metabolism and something clearly related to patching or enucleating only one eye. Well, then we began to think, well, this sounds very much like what Ayub and Wiesel have been talking about. So we looked up Ayub and Wiesel's papers from 68, 72, and they had a artist's reconstruction of the ocular dominance columns, but they were nice rectangular slabs with smooth borders, and these didn't look like that. But it sounded very much like that. So I had met Wiesel at some meeting in Japan, so I took the liberty of sending him a photograph of the autoradiograph of the visual cortex with these stripes, these columns, with a note saying, you might possibly be interested in this. And I didn't hear for about a month. So I thought, well, I guess he's not interested. Well, about a month later, I got a call from him from Boston, excited, very much interested. He'd been away in Europe, and he had just returned to see the photograph.
picture, yes.
He was very excited, and he and you will send Mike Stryker down to learn the method in our lab. What it did do was, for the first time, illustrate the shape, the extent, not the idealized shape, but the real shape, the distribution, and of the columns, of the ocular dominance columns. A side thing came out of it. We noticed that in the deep calcarine cortex, contralateral to the patched eye, there was a spot that acted as though it didn't know an eye had been patched. But at the corresponding spot ipsilateral to the patched eye behaved on the autoradiographs as though it thought both eyes had been patched. And when you work out the neuroanatomy of it, then it became very clear. We had found the cortical representation, the cortical loci of the representation of the blind spots of the visual fields.
That's fascinating.
Yes, right,
yeah.
right. Yeah. Localized it entirely.
By a functional method.
Right,
yeah.
right.
Yeah, it's a beautiful study because it ties in so many different techniques. And then, of course, in the memory experiment, it tied in the anatomy of the two systems, the spatial...
Collaboration with Kathy Mako and Mark Michigan, yes, showing the inferior temporal cortex, the extent of visual function there.
Yeah. And object function. This brings us to something that's, I think, very, very interesting, and that is in the recent progress in hormone research, 1983, you review your previous work, and there's one table which I'd seen previously also, looking at the local cerebral glucose utilization in the normal albino rat and the monkey. And in that, there's quite a striking difference in the regional distribution in the different parts of the rat brain and the monkey brain, with the monkey brain having a much lower rate of consumption as compared to the rat. And you explain that by saying this is related most probably to the packing density, because when you look at these figures, it looks as if the monkey brain is somehow more efficient than the albino brain. Now the question is this explicable in terms of the packing density alone, or is there some other factor, because the packing density is related more to the number of the neuron glia index, where you have, and the glial cells themselves are active.
Right. That packing density was just a hypothesis of an explanation that it has not been proved, and I'm not sure I know for sure why the little brain of the rat has a higher rate of metabolism, at least between two and well, it's more than two or threefold more than the monkey, and I don't know why. And smaller animals should have a higher cerebral metabolic rate than.
This seems to hold for general body metabolism.
Yes, it does, but I don't know why the brain should partake in that. You know, that's a different issue.
That's right.
The brain is not involved in temperature regulation or distribution of heat, so I'm not sure. But the packing density and the fact that the rat has less white matter than the monkey may have something to do with it.
But when I tried to look at it in terms of the packing density, I couldn't get a very good correlation. Also, the human data also suggests that the human brain, in a sense, is even more efficient than the monkey brain.
Yes, the average glucose utilization in the monkey brain on the, in most series is somewhere 35, 36 micromoles per 100 grams per minute. The human brain is about 30, 31 micromoles per 100 grams per minute. And the cat is actually a little more than the monkey. But less than the rat. Yes.
So it fits very well on the phylogenetic scale.
It does, yes.
But the exact explanation of that remains
we don't
an open issue.
We don't know. Actually, we've learned something else more recently in the last few years. That the functionally dependent changes in glucose utilization are mostly due to what goes on in the synapses and not in the cell bodies. Dr. Catacaro, who used to be in our laboratory, did these following studies. She stimulated the sciatic nerve in a rat And she measured at different frequencies, electrically stimulated. And she measured the glucose utilization in the dorsal root ganglion, which has no processes, no synapses, just cell bodies. And then in the dorsal horn of the lumbar cord at the same level as the dorsal root ganglion that she examined. And what she found was that in the dorsal horn, where the nerve terminals of this afferent pathway end, the primary input, the nerve terminals end in the dorsal horn of the spinal cord, there was a practically straight line relationship between the spike frequency, the frequency of electrical stimulation, and the glucose utilization. The dorsal root ganglion has a glucose utilization that's steady, but it was completely unaltered by the electrical stimulation. And she has now done some experiments where she antidromically stimulated the ventral root and looked at the motor neurons in the ventral horn of the spinal cord and found similar effects, that the cell bodies do not seem to respond to the stimulation. This would suggest that the cell bodies have an energy metabolism, but it may not be directly related to functional activity. It may be just for maintenance. But the action potentials that are responsible for the increased glucose utilization are those that occur in the synaptic terminals, in the axonal terminals, which raised an interesting question. We, as previously indicated, had evidence that spikes should lead to glucose utilization, increased glucose utilization, because of the potassium coming out and sodium going in, turning on the sodium-potassium ATPase. And cell bodies have sodium-potassium ATPase. Why, if there are spikes in the cell bodies, don't you have an increased glucose utilization then? So we talked to neurophysiologists who record with electrodes and asked, do cell bodies spike? And they said, of course, they've recorded many times. Then we talked to the patch clamp people. And the patch clamp people said, oh no, the pericardial membrane is not excitable. So we said, well, how in the case of the dorsal root ganglion, how would you get the impulse going past the cell bodies and go to the nerve going to the dorsal horn if the cell bodies don't spike? And they said electrotonic conduction, not spike. If you examine the cell body membrane, there are very few sodium voltage-dependent sodium channels that won't spike. And so if that's the case, then that would explain why cell bodies do not have action potentials, the glucose utilization is not increased, while where the terminals are, they are. So that's an area that we're really interested in pursuing further.
This gets to one of the larger questions from which comes out again from your work, and it's based on a lot of the earlier work where you have shown that with mental activity and the sort of work which the mind, when everybody talks about the brain, they think of the brain's work as mental functions, and yet we find that when that kind of function is looked at, the energy consumption is redistributed in the brain, tremendous regional organization. But overall, there's either no increase as measured, or if there is an increase, like in recently with oxygen studies in Sweden, Roland and his group mentioned a total overall increase of 10%, which again is a very small increase.
Yes, this is an area currently of intense debate. And it was started from the Washington University School with Fox and Rakel.
Yes.
They had found with their PET studies, using their methods for measuring blood flow, in which they use O15 labeled water, and the oxygen consumption method using O15 labeled molecular oxygen, and a carbon monoxide scan and a blood flow scan, and all three. They had found that with a sensory stimulation that they were able to get increases in blood flow of, I don't know, 50% perhaps, locally in specific regions. But when they measured the oxygen consumption, they couldn't find any increase or minimal increases. And so their first interpretation of that result, which I heard for the first time in 1985, at a meeting of the International Society of Cerebral Blood Flow and Metabolism in Sweden. The first interpretation was that increased local functional activity increases blood flow, but does not change energy metabolism, period. Which didn't make any sense because we had all this evidence that glucose utilization increases.
Yes.
But they hadn't measured glucose utilization, so they had generalized to energy metabolism. I think probably as a result of that meeting, because it was such a surprising report and caused, as you can guess, a lot of hostility.
Exactly.
The suggestion was taken by them to, well, measure glucose utilization. So they went ahead and measured glucose utilization and subsequently. And then they found that glucose utilization was increased almost the same as blood flow. But oxygen consumption wasn't increased. So they changed their interpretation to say, well, it's glucose utilization is increased and blood flow is increased because it's only that the functional activity derives its energy from glycolytic metabolism and not aerobic metabolism. So that's the current state. And there are a number of people who are all excited about this. I personally have great difficulty of accepting it. One reason is that I have a background in biochemistry, and I've done a lot of work with mitochondria and oxidative phosphorylation and so on. And I cannot conceive how it's possible in the presence of adequate oxygen to increase glycolysis without increasing oxygen consumption. The same cellular changes that turn on the glycolytic pathway by regulation at the phosphofructokinase step are the same changes that will turn on electron transport in the mitochondria and lead to increased oxygen consumption. I think, and and this is not if I have no proof, I suspect that the problem is that the method for measuring oxygen consumption is defective. This is the method that requires 3 scans. And you have to take the results of 3 scans and put them together to come out with a single measurement of oxygen consumption. And one of the scans, the one about the one with the carbon monoxide, to determine how much oxygen is bound in the hemoglobin in the blood in the tissue, is I think the one that may be the source of the error, that they may overestimate the amount of oxygen is bound on the hemoglobin. When they subtract, they lower the oxygen consumption, they produce a ceiling. The reason why I suspect this, another reason why I suspect it, is that in their paper in Science a year or 2 ago, they gave values for the oxygen to glucose ratio consumption by the brain in the normal resting brain of about 4.1. And when they stimulate, it went down to about, I think, 3.6 because the glucose was stimulated, but the oxygen wasn't. Well, 4.1 is ridiculously low. For 50 years, people have been measuring the ratio of oxygen consumption to glucose utilization by model-independent methods that involve only the chemical measurement of glucose and oxygen in the arterial and cerebral venous blood, and looking at the ratio of the AV differences. There's hardly anything that can be wrong with those methods.
Very direct measurement.
Right. And it's certainly not dependent on any complicated models and computations and calculations. And they have almost universally found, on the basis of results of a review that I had to do of this area a number of years ago, almost universally found that this ratio of oxygen to glucose is between 5 and 6 to 1. The median value was 5.5 to 1. The group from Washington University in the resting brain had found 4.1 to 1. So there must be something wrong with one of their measurements because, and it's not surprising since their methods are so dependent on the validity of models that may or may not be, usually are not very perfect.
There's one final question I'd like to ask you, Dr. Sokoloff, and that is about the very intriguing issue that is raised when you hear people, very good scientists like Dr. Chance, who's when asked what is the work of the brain, he says, well, it's pumping ions, which of course is one of the very important things that the brain does. But when we think of the brain and the mind, we think, of course, of mental activity and consciousness. And that is not answered by that question, because we obviously have to relate it in some way. How do you approach this problem?
Well, the physical work that the brain does that takes energy is pumping ions. And as you say, this does not translate into consciousness and thought and motion and all that. I think those higher levels of function still are dependent on the pumping of ions, but they are dependent on where the pumping of ions is occurring. And that's why I think methods for localization in the nervous system are so critically important.
Do you think that if one follows the localization approach, that we can indeed solve the problem of consciousness and thought by basically assembling in other words, discover the laws at the level of organization of the modules as well within the brain, that is compatible with what is called nowadays the computational approach to the mind, which is a favorite of artificial intelligence people, mathematicians, or will we need understanding also at a higher level of new laws at the level of the organization itself, which again will use these modules, but with their own laws rather than reduce down to the physics and chemistry as we know it?
Well, I don't know that we'll ever solve it, but we certainly can approach it closer and closer asymptotically. Take an infinite amount of time to solve it thoroughly. I have, on occasion, wondered how it is possible, even if we were able to figure out all the synapses that are involved, all of the localization involved, how spikes will translate into the feelings of consciousness, the qualitative aspects of it. You know, I feel this. It has a certain feeling that I'm aware of and conscious of. I have a little bit of difficulty conceiving of how localization of spike activities and neurotransmitter releases results in this conscious sensation.
That's right, which only you know.
Yeah, and nobody else knows.
You can assume that somebody else knows,
right.
But nobody will know what you feel.
That's right. It's unique. Consciousness is unique to each of us.
Well, that's at least we know we can be comforted by the thought that the greatest minds in philosophy have still not solved this problem of the qualitative aspect, the qualia, the problem of qualia, the philosophers battle with.
Right.
But hopefully we can maybe approach it, as you said, step by step. Thank you very much, Dr. Sokoloff.